It's not easy. Try something. And then whatever comes out of the discrete choice experiment, of course, it's going to have to be trials in some way, shape, or form as well. That's just going to be people's opinion about what should work for my
Do we need to press anything else? This is recorded now. Yeah, I think we're right.
Sorry?
Did you unmute? Yep, we're unmuted. We're recording, yes. All right, we're running a little bit late, so we'll get going. Hello and welcome to everyone who's online, and to everyone who's in the room, and to our March Journal Club. And I'm going to introduce Lydia Upjohn, who's one of our research fellows who's been working with us in VIDS since the partnership grant, actually, so quite a number of years now. And she's going to have a chat to us about first article for Journal Club.
Thanks, Kirsty. Oops. Okay, so I was thinking about what would be a good article to review from the general literature for Journal Club today. And so I thought we would pick this article, which has had some press and has also been published in The Lancet. And it's a really enormous study in the UK. And I thought it was had some really interesting general concepts in it to think about as well.
Is that the Sally Davies?
It's the Sally Davies, which is one of the things that I'll talk about. So the article's called Provision of Social Norm Feedback to High Prescribers of Antibiotics in General Practice, a Pragmatic National Randomised Control Trial. And so this introduces the whole idea of something called social norm feedback, which is basically another word for peer pressure. And I just thought that was this looks at general practice and what can happen in general practice, but I thought it was a good concept to think about in all the other areas of stewardship, whether it be aged care, whether it be hospital practice, as a way of influencing people's prescribing. And this is a big recent study. So this study was done by something called an organisation called the Behavioural Insights Team, and it's a UK-based funding organisation. I saw a behavioural insights team. I thought they have to come and see my children. I need them. But they actually do what's called it's a social purpose company, and it looks at ways to make public services more cost effective and introduces realistic models of human behaviour to policy, which I thought was a really interesting kind of mandate for your organisation. And these are the kind of studies they've done in the past. They've done studies looking at SMS reminders in reducing missed hospital appointments, a trial, and seeing how much money they could save by doing that. And they've looked at whether they can increase voter registration by running lotteries in England. So quite interesting kind of big studies looking at human behaviour. So they talked about talking about feedback in antibiotic prescribing. We've had mixed results in studies so far. And feedback is often used as one aspect of a complex intervention. And the question here was, can complex interventions feedback be scaled successfully to general practice?
It's just...
okay. All right. Thank you.
Sorry.
Might have to keep going with that view. Sorry about that. Okay. So what's social norm feedback? And I was just looking up at what other things they had looked at in terms of, and called social norm feedback and what it is. So it's presenting information to show that individuals are outliers in their behaviour, and it leads them to adjust their behaviour towards the social norm. So other studies that they've done in this area is looking at healthy food choices or reducing binge drinking or reducing gambling, that kind of behaviour. So this study design was a 2-by-2 randomised control trial. It recruited GP practices in England, and it used prescribing data sets from detailed prescribing information that is available from primary care in the UK, and it's only really been available in the last few years, this detailed prescribing information. So this is the flow chart looking at their study. It's a little bit hard to read, but essentially they looked at all GP practices in England who issued prescriptions for any antibiotics in the month before the studies launched, and they very purposely excluded, so that's the green box on the right, the top very highest prescribers. So the top 5% were cut off because they said there's something about these practices that's different, and that was to prevent inappropriate communication with practices who were different from the main population. And they excluded the practices who were not in the top 20% prescribers, so 80% went out at the bottom, and they just looked at that 20% band at the top, which they thought was the optimum trade-off between statistical power by widening the sample and diluting the power of the social norm messages. Yep. Just keep going. So this was the letter that they sent out. So they had 2 interventions. The first intervention was called the feedback intervention. And in the feedback intervention, they said that they sent out this letter to the group that was in randomised to that part of the trial. So I'll just go back. So we're flowing along here. They excluded the top prescribers, and then they had controls, and they had the feedback intervention group. And in the feedback intervention group, and then they put them in again, and then they divided that up into the second intervention controls and those who went on the second intervention, which I'll talk about. So they had 2 different interventions and controls. So this was the first intervention. This was a letter that they sent out. They identified the top 20% and sent out this letter saying, compared to other practices in the local area, you are in the top 20% of prescribers. A little bit hard for me to read from here, but then they said we recommend that they've given them some options for ways that they can, simple ways that they can decrease their antibiotic prescribing. And then they said they signed it from this high profile figure, Sally Davies, who's the Chief Health Officer. So stating that comparing to other practices in the local area made social normal information more specific, that was the idea that it was be very effective because it compared to other practices in the local area. It increased credibility because it was addressed from this high profile figure, and it gave 3 feasible actions that the participant could do, and it was addressed straight to the doctor in the practice. Also, the feedback intervention group were also provided with this target leaflet, which reinforced the message of the letter because it acted as a method to support delayed or reduced prescribing. And I actually hadn't seen this before, and I thought it was really good, particularly because I was looking at sinusitis and cough and bronchitis, and they give you days of, I think it says the average length is 18 days and 21 days. So it gives patients an expectation that they're going to be sick with a bronchitis for that long, because I think patients often get to about a week or 2 weeks and say, oh, I've just had enough and I'm going to go and see my GP. But it gives them, look, I've just got to wait a little bit longer and it'll probably go away. And it gives them kind of a clear message about when I should actually go back and seek medical attention. And it provides this option of a backup prescription to be collected from my GP after so many days. So the idea of I've gone away from this consultation with a prescription, but I'm just going to wait this 21 days or 18 days and see if I get better. So I thought it was really simple and really good. So then they randomised people, the groups, the practices, to another intervention, which was called the patient intervention, and this was as follows. So they mailed out posters and a leaflet to the practices, not to the specific GPs, just to the practice manager in the first month, and said, here's some patient education material that you can use in your practice. And this was the poster that they used. A fact about antibiotics you may find hard to swallow. I can't quite read all the information from my little screen, but if you can read up there what it says. And they've done a lot of qualitative research on this poster and found that the most effective messages would be those that link unnecessary antibiotic use to future personal consequences arising from antibiotic resistance. So people don't want to know that this is going to affect the whole population. They need to know that it's going to affect their children or grandchildren. And then they also included this leaflet. And to me, this leaflet looks very similar to the previous target leaf thing that was sent out to the GPs. So I didn't think it was specifically a very good patient-focused thing, because it was something that was just sitting in the waiting room that they could potentially pick up, I suppose. And the idea was that there may be pressure from patients to prescribe antibiotics, and it was intended to promote the attitude among patients that if antibiotics are taken when they're not needed, they're less likely to work. But it's got a similar message to their target, that their illness is going to last an X number of days, and this is what they should do. So the control group did not receive the letter, and if the intervention had been shown to significantly reduce prescribing rates, they plan to send the intervention at the end of the month to all the other prescribers. So their outcomes, they looked at their primary outcome measures were the rate of antibiotic items dispensed per 1,000 population, and their secondary outcome measures were if there was a significant difference between the intervention and the control. They were going to estimate the total differences in the number of antibiotic items dispensed and the effect on direct prescription costs. And this 2x2 design that they used allowed them to look at the aggregate effect of the two main interventions together and then the effect of all the 4 experimental groups. And they powered the study to detect a change in the dispensed antibiotic rate of 0.48% with 80% power, and they controlled for patient demographics, local area prescribing rates and previous prescribing trends. And they used the practices prescribing levels 12 months before each of the months in the study period as a control to see if there was any change. And they interacted the two interventions to see if combining the interventions had a statistically significant additive effect. So these were the results of the feedback intervention group. So that's the group where the GPs got that letter and the target leaflet. So the rate of antibiotic items dispensed per 1,000 population was 131.25 in the control and 126.98 in the feedback intervention. So there was an absolute difference of 4.27, which was statistically significant, and so it's a 3.3% relative difference. So in the feedback intervention, it reduced the antibiotic prescribing by 3.3%. So small but significant. So here you can see highlighted at the bottom there, this is the controls and this is the intervention. And they pulled the data from all of the months. So they first distributed the letter in September. They had a significant result in all of the months up to the end of the 6-month intervention. And they pulled the data, and you can see that the incident rate ratio, which is the number of new cases of a condition, i.e. an antibiotic item dispensed, was reduced by 0.967 with a significant a p-value of less than 0.0001. Next. And this is just a graphic to show, give you an idea of what that prescribing rate was like. So this is where this was pre-intervention, and then they posted out the letter, and there was this split in the feedback intervention versus the control group. It looks, but it's small, but it's there the whole time until they posted out the letter to the control group as well in this month, and then they came back together at the end. So the rate at which antibiotics were dispensed in the feedback intervention group was significantly lower than in the control group for every month of the study period. So that patient-focused intervention, which was the second one, which was the poster and the leaflet sitting in the GP practices, showed that the rate of antibiotic items dispensed per 1,000 population was 133.98 in the control and 135 in the patient-focused intervention. And basically they showed non-significant results. So when they first posted out the poster and the leaflets in that first month, they actually had a significant increase in the antibiotic prescribing rate for that first month, and then it went, the rate at which the antibiotics was prescribed was non-significant. There was no difference. So the results of all the 4 groups are here. So in the antibiotic prescribing rate was significantly lower in the group assigned to the feedback intervention only group compared with that received neither, which was statistically significant. In the patient-focused intervention, it produced a significant increase in the prescribing rate in the month it was delivered, but its effect was not significant for the whole period. The prescribing rate was significantly lower in the group assigned to both interventions compared with the group that received neither. So you can see this. So in the first, it's just to show that in the first month that they posted out the poster and the leaflets in the patient-focused thing, that intervention caused an increase in prescribing rate, and then it went back to normal. But when they looked at both of them together, it was still significant. But I suspect that was mainly the feedback intervention working. This is a little bit hard to see because of the closeness of the lines. But essentially, the rate at which antibiotics were dispensed in the feedback intervention group was significantly lower than the control group for every month, which I said before. So in conclusion, a male-based intervention incorporating social norm feedback on high antibiotic use can reduce use over a 6-month period. There seemed to be no significant effect of the patient-focused campaign material. In fact, it seemed to increase risk of antibiotic prescribing. And the difference, they looked at the difference of 3.3% and said that it saved 73,406 antibiotic prescriptions, which is actually a lot when you think about it. So that's what 3.3% equated to. So the limitations of this study were it didn't look at outcomes apart from prescribing data. We don't know which conditions did the reduction in prescribing occur or what antibiotics, which would have been interesting to know. The intervention was practice-focused, not individual-focused. There may have been some cross-contamination because doctors may have been working at multiple practices around the local area. And we don't know, the intervention looked at delayed prescribing, but we don't actually know what the rate of delayed prescribing was. And we don't know the effects of repeated messages. So this was one intervention where they sent out one letter and it looked at 6 months. But I guess the question will be, what happened if you did that again? Would the effect peter out? Is this just something that you can use one-off, or is it something that you can use again and again? And the other thing is, it's difficult to test out what was social norm behaviour because, and by that I'm talking about I'm in the top 20% of prescribers, and what is useful because they included a rather good leaflet in there that the GPs could use, or was it because Sally Davies signed it? I don't know. But I suspect that knowing that you're in the top 20% would be a little trigger And then the leaflet's actually quite good and quite useful for GPs to use. But how much was the influence of each of those things, the study couldn't tease out. My thoughts on the study, look, this could only be done in the NHS, where they've got a lot of prescribing data. The GPs are all on, essentially employed by the NHS. There's a lot of public reporting. People are used to kind of scare messages and scare campaigns. You are being watched. I don't know how well it would fit into the Australian context or the American context, where we're more individualistic perhaps. We've got a different set of remuneration, kind of a different set of GP remuneration. But I thought the intervention was very appealingly simple, and I really liked the treating your infection leaflets. The other element was the accuracy of the prescription data. They didn't go into any detail about how they collected that data, but, they said it was prescribed data, but we don't know about dispensed and we don't know about what was actually taken. And it's really still in the fax machine era intervention. I think now we're more likely to aim for a process where practices log into a My Antibiotics GP portal and have regular updates on prescribing rates, and little alerts go off when you hit the top 20%, and you get a email in your inbox to the practices manager saying, you've reached the high prescribing rates. I don't know. I think this is a useful intervention, but maybe there's ideas from this that we can bring back to how you would, bring in a more modern kind of technological way to feed back to people. And then I was thinking about how do we test or use the results in other areas for hospital practice, knowing that you're an outlier in your prescribing and that you get anonymous feedback that you're an outlier. And I was thinking about what the social norms were in hospital prescribing. So empiric vancomycin for any suspicion of staphylococcal infection or hospital-acquired infection, 24 hours of kepazoline practice post every operation, oral prophylaxis for 5 days in lower limb skin grafting, things like that are social norms in hospital prescribing. They're things that are learnt from influential registrars and consultants and passed on to junior prescribers, and they take that through their prescribing practice. And it's also a matter of looking, the other element of a social norm I think in the hospital is when registrars and consultants look at what the other units are doing, and they want to kind of know that they're not prescribing less or not doing more for their patients than the other chaps down the road or on the other ward. They want to. So I think there's something useful that we might be able to think about in knowing that doctors really just want to conform.
I posted a thing on Twitter, I think it was yesterday, about Reeves with this Indian cardiac surgery hospital, because there was it was an article about what are the highest performing hospitals. Now, I'm not talking about general practice, but what are the characteristics of the highest performing hospitals, and they describe some of the characteristics, and they gave this case study, and it's actually in India. It's a cardiac surgery hospital. They carry out the average Australian cardiac thoracic surgeon, I think, performs like, I can't remember, 150 a year or something, but they do like 1,000 a year, each surgeon, just like pumping them through. But their infection rates and complication rates are half of what are the best rates elsewhere in the US. And that actual hospital, the way it works is that at midday every day, there's a profit and loss statement posted to every doctor in the hospital. They are continually compared to each of their peers as to what their complication rates are. So everybody knows what every surgeon's outcomes are. And so it's this real push, like this sort of, like real
a driver, a push to.
I don't know about the profit and loss statement, but I just thought it's just fascinating. A, the throughput is so incredibly high, and B, that their real outcomes are so good.
Yeah, so I thought the message from this study was that we might be able to use this, feedback.
Well, I think this is Medicine Insight. That's what they're trying to do, I think, aren't they? Medicine Insight give the GPs feedback. I don't know how often.
Yeah. 7 months.
Oh, no, once.
7 months.
Where have you prescribed compared to
It's only certain antibiotics.
respiratory tract infections?
And I really like the target leaflet. I thought that was great. Yeah. Okay.
Thanks, Lydia. Now we should just check if there are questions too from anyone out in. How are we going to see them?
So you just, questions,
Yeah. So we know it started late, the sound, audio went.
Just explain that we've just got to
Okay, now we're yes, we can see that there's been problems with the slides being too small. So we've got right up here seeing if you can try and figure it out for us. We're sorry about that to everyone out there. We are trying to fix it. In the meantime, will we invite Helen up? Do you want to pop your slides in, Helen, while we prepare? Thanks, Lydia, that was terrific.
So is the issue relating to where the questions?
We don't need the questions sorted.
Oh, okay.
Are we doing this
We
slide
need to fix this slide size.
Maybe if you can.
I think she
Apologies everyone, Phil's gone up to Sydney today and he's normally a technical person, so we're having to struggle with this system on our own.
When I presented the other day, I didn't have the 4 screens on that screen either. It was just the presentation. So,
do you know which button to press?
well,
Can you just upload one?
Yeah.
Just click this a moment.
Which one?
I'll just make this one big. Yes, like
That's what I know.
it's still doing that, so that can't just stop this blue.
yeah, I didn't have that screen.
Oh, yeah, it's working there. Oh,
Yeah, it's okay.
it's not working.
Yeah, it's all
But as long as it's on.
Hang on, there must be multiple displays.
Can you go to here?
Check mine.
Sorry.
Okay.
And just go to here. No, just yep. Screen sharing. Go to my. Yep, and go to my screen. Have you opened one of those screens again? One of the presentations into Stewardship.
It's right up there. What have you got?
No, it's still in the floor.
It's still on the floor.
Yeah.
Settings here.
Can you just do mirror show instead of presenter view up at top?
Can you come and show us? That's it. All right.
So that's mirror show, the 2 displays. Okay,
Can you have that up and down?
what's that?
This one.
Oh yeah, or just one.
Is that working now?
That's
Is that working?
Yeah.
That's working?
Yeah. Okay, and then you can.
Is that working?
You can push that over there.
Well, that's the wrong,
That's better.
the wrong presentation. Helen, have we got, okay. I think we know what we need to mirror show.
No, but that's not normal. That's not what I normally do.
Oh, okay.
Yeah, so that's going to fix it. That's not normal. Okay? I will be like, oh.
Sorry.
So let's see back here.
At the uni, different setup. You'd think they'd put the standard setup in here. Anyway. You don't even know where it is.
I can spit.
I'll let you finish that.
Oh,
Oh, there it is.
good.
It's all right. This one? It's really good.
Except.
Oh, the ribbon's gone.
Yeah.
Close it.
Is that it? No. No. Where is it on desktop?
It's on
It's on
I
there.
can't see it. Oh, there it is.
Yeah.
And it just should be saying coli.
infection. Take it off.
Okay. And we just need to make this a slight shape. I just want to make sure I've got that mirroring shape. What's it look like online? You can't read that, can you? Oh, God. Okay. Now?
That's what
Yeah.
I think we'll go.
Okay.
You won't be able to see anything.
Now, what did we do before?
It was that mirror.
Mirror. And there's something here as well. So have a look down. Have a look down.
Here. Okay, we've got it fixed. So I'm Helen, Billman-Jacobe. I'm from the Faculty of Veterinary and Agricultural Sciences, and I'm involved in the animal stream. I looked for a paper that had an intersection of animals and humans, and the story that I'm going to tell you is about a family and an important member of this family, the E. coli that live with them. And the story goes over a number of years, and it's a series of papers culminating in looking at some of the mutation changes that happens in the E. coli. So the concept of clonality is important in these works, and I pulled the quote out from the final paper, which explains that E. coli is a clonal species, and that clone can live in isolation for very, very many years. Although in the strict sense of a clone being descended from a single recent ancestor is often lost in E. coli because the chromosomes rearrange through a process called recombination. So genetically there's diversity, although they've descended from the same parent. In these papers they use the term clone to refer to isolates that have a clear, most recent common ancestor in terms of cell division, because these are just dividing by binary fission. And we're able to accommodate this problem of genetic plasticity of recombination in those analyses. And the shorthand we use for the ancestor is the most recent common ancestor, which is often a hypothetical ancestor that has never been isolated, but we're able to track back what its genome would have looked like. So the story starts with this family in 2005, comprising of a mother, father, 2 daughters, the son, and the family dog. And the mother of the family had an acute episode of cystitis, and that she was promptly treated effectively with fluoroquinolone. And I only mentioned antibiotics twice here, and that's what was used to treat the 2 cases of infection. The reason I chose the paper was to show how in this community organisms that can cause disease are shared throughout the members. So the mother had one sort of E. coli in her urine of more than 10 to the 5 CFU per mil of E. coli, and at that point they got the family to agree to participate in a study, and fecal samples were taken from all the household members, including the dog, and then they followed up with subsequent samplings at week 3 and then week 7 to 9. This is back in 2005, 2006, so this is before we're just processing them through easily to whole genome sequencing. The analysis they are doing here
to type the different clones is based essentially on some PCR techniques called RAPD, which give you a profile and you can work out genotypes. And then they can take representatives of the genotype and further discriminate their relationships on an agarose gel-based method called PFGE. So they're able to group them broadly. So what they found among the family is that there were 7 unique E. coli clones present, and that when we look at the different hosts and the different sampling times, we can look at different diverse colonisation behaviours of the E. colis. So clones B, C, F and G were only isolated at one time, and they were considered to be non-persisters. A persister needed to have been isolated in multiple hosts and on more than one occasion. We had 3 persisters, D, E and A. D and E were recovered from 3 hosts, and clone A was isolated from 5 hosts. And they depict these sampling in this type of graph. So here we've got the various hosts, the type of sample specimen taken, and then the clone and its name. The width of the bar relates to how many colonies were isolated from that sample. So this, which was a pure E. coli population, has a big bar, and here a few colonies of this type was found in the subsequent sample. So what we see for clone A is that it's present in the mother, the father, it gets resolved in the mother, persists in the father's sampling. We see it appearing in the daughter. This is a missed sample, and we see that the son and the dog also having this clone. So when they are able to look at this clone genetically, it is most associated with a type of E. coli that's previously been characterised, associated with cystitis. And so it's a virulent E. coli, extra-intestinal E. coli. The other persistent clone, clone D, isn't present in the mother, but we see it in the daughter on 2 occasions, the other daughter and the dog. And again, this clone is a virulence-associated extra-intestinal E. coli, and the most common E. coli in the to compare it with is one that's been isolated from a case of pyelonephritis. So we've got these 2 persisting clones. That was the end of the first paper. But then in 2007, the dog experienced a urinary tract infection. And it was, again, promptly treated and effectively treated. And its urine sample had, again, large numbers of E. coli. So the family, I thought, was remarkably cooperative. And I suspect that perhaps one of the authors may have been related to these people. However, so again, they all consented to sampling, and so again they took samples concurrent with the dog's UTI, and then 1 and 11 months later, giving us a 3-year period of study, which makes this study unique as far as looking at associations of E. coli in a controlled environment and looking at host transfers. They used the same techniques to type these and work out the clones, and at this stage, they're able to pull the data in the 7 in the 2 studies, and we end up with 14 sorts of clones. Individual hosts had up to 6 clones, and 3 or 4 of them were shared by all of the hosts. So there was a fair bit of sharing going on. They did have a look at some of the interactions between family members and diet and sexual contact to see whether there was a ready explanation as to whether there was sort of an external pressures here. The dog was vegetarian, the dog ate commercial dog food. The daughters, interestingly, had virtually no contact with the male members of the family. So the dog is the central contact point for everybody. So what we see overall is everybody shared at least 1 to 3 clones with every other member of the household, and that we had some long-term persisters, including the dog's urinary tract infection clone. So if we use their same type of scheme, but now we're taking the first study and extending it over the subsequent samplings, what do we see? So this is familiar. This is the mother's UTI infection, which ended up in everyone else. We've got our dog's UTI, which now we see appearing in the sons and the father, the daughters, and the father, and ultimately the son as well. But in this case, the mother didn't get it. However, the mother shares clone H with the dog and nobody else. So in trying to look over time as to where the various samples are going, they're represented in this, where each of the colours represent a different clone, and I've stuck to red for clone A and purple for clone D. But we see essentially the clone A, which was the mother's infection, circulating in this triangle, and then it goes from the father to the son, and then the son to the second daughter and the dog. And then when the dog got sick, this is clone H, which the mother and dog shared, but this is the dog's UTI infection clone, which previously existed here, but now it's going between the daughters and then around this rectangle with the father that the mum missed out. So at this point now they've got a whole lot of clones, and time has moved on, and they're able to do whole genome sequencing to get a more detailed analysis of what's happening with the clones within the family, which is what they do in this paper. So when they do a whole genome sequence, you look first of all at the core genome, which are the genes that are shared by virtually all of your isolates. And then there'll be other genes that aren't shared, and you don't use those in looking at the phylogenetic relationship, you just use the core genome. So the most stable part of the genome. So they looked at their clones, principally clone A and D, and how it related to the other E. coli genomes that were available publicly at the time, and they're able to see that clone D falls into this cluster, which is distinct from this cluster, and so the distance between that cluster, which you can follow the line lengths, shows a distance of evolution of relatedness. So what we're able to conclude from that is clone A and clone D are not related. Then if we look at the relationships of just clone D, phylogenetic the relationships of it and all the isolates of clone D over time. So they're colour-coded according to the host, and I've put them here there so it's easy to sort of to discern. The most important host, of course, is the dog, which is blue. So the length of these branches represents the number of mutations that have occurred since the most recent common ancestor. And the isolates, each of these represent an isolate, so this is a daughter clone. So we have essentially two branches where this goes into this variant, this genotype of clone D doesn't persist. This one goes on to have further mutations and persists in two branches. There's what we call the dog branch, where we've got this isolate which subsequently mutates and is re-isolated from the dog. And then we go off on this branch, and I6 then has further mutations. We've got 3 isolates of that from the dog and 1 from the son. So these are genetically identical, and this one's the urinary tract infection from the dog. Because they're all identical, we're unable to say which one gave rise to these subsequent genotypes, but it's because this is essentially clonal. So what was unique about this is that it was an opportunity to look at genetic change over time. Now, this doesn't say that the organism is becoming more virulent or more resistant. It's just looking essentially at mutation rates. Although they were able to say that they think that this branch may have acquired some fixed mutations related to host transfer. So it was a dog-adapted strain because they never see this in anybody else and it never reappears in a human. So that's the hypothesis that it's host-adapted. This is their other way of representing the data according to time. It takes a little bit to absorb this one. Each square is a real isolate, and each black circle is a most common recent ancestor that can be inferred from the genotype of the real isolates. So, for instance, this isn't an isolate, but we know this strain and this strain have some things in common, therefore its ancestor must have had those mutations. Each of the arrows refer to mutations acquired since derivation from the common ancestor, and they're colour-coded as to whether they're in genes. So if they're in non-coding regions, they're blue. If they're synonymous, which means that there's no change in amino acid sequence and therefore will have less evolutionary pressure, they're in black. And if they're non-synonymous and that they change the protein sequence, they're in red. The aqua, I think they call it, circles represent a host transfer. So the host transfer can be from human to human or human to animal. And in mapping this, because they've got extremely good sequence data and they've seen every single nucleotide polymorphism that was possible, they're able to do this fine detail evolution and see how things change over time. And then they can ask some questions about how this would compare to a controlled environment. So this is a natural environment. Equivalent studies have been done with E. coli, where they've had E. coli in culture for 20 years under the exact same culture conditions, representing 40,000 generations. And under those circumstances, where you've got consistent selection pressure, they're able to work out evolutionary rates, which are incredibly small in E. coli and in bacteria in general. So we know they don't change very fast, and they're able to calculate back from what they see in this natural environment what's actually happening. So the conclusions were essentially pointing out that this was a fairly unique study, that they had a whole lot of isolates and genotypes. The mutation rate that they're able to work out was 1 fixed mutation per year. And that's about 6-fold higher than expected. And the expectation is set from this 20-year-old longitudinal culture experiment where they know what would happen in a constant environment. Now, as a bacteriologist, I still marvel at that because you get the feeling, you get the impression that bacteria are adapting and changing all the time. It's really the huge populations that we're selecting rather than them actually mutating at a great rate. This study was, again, it was a very useful type of E. coli, fortunately, to look at, and that it wasn't having a lot of recombination. The chromosome was stable, and they weren't having new DNA coming in, like mobile elements coming in and causing that plasticity of genome. So it was a fairly stable chromosome as well, which uniquely gave them the opportunity to look at mutations. In this setting where you do have, say, plasmids arriving with a whole lot of antibiotic resistance genes and transposons and integrons and a big inflow of DNA, coupled with selection, this could have evolved or there could have been selected to be a much nastier E. coli, but it was certainly interesting that whatever happened at the earliest point of treatment was going to have an effect on that family well and truly down the track. So I guess it's a good argument for effective antibiotic use. That's the study.
Helen, this concept of the microbiome, it sort of seems to hit the headlines in everything now. Nature, Microbiology, every week there's more microbiomes.
Yes.
And I thought I'd present a couple of papers at the conference coming up, and I'm trying to find out, clearly this is because of whole genome sequencing that it's a whole new, there's relations with all sorts of things, cancer,
Whole gut health of the microbiome.
allergies, hypersensitivities, all sorts of things. It's obviously clearly going to be the next big thing, the new black for ID. What's your take on the current stuff that's coming out?
So I think that, like that study, and it is fairly old, where you just look at one thing you picked out of the fecal sample and really study it in detail, and now we want to know not so much which organism is carrying the particular gene, but what's in there. And from our own work, if you're particularly looking at just the pathogen and you're ignoring the commensals or ignoring other organisms, but next week those genes will have transferred. So I'm more interested in not necessarily the bacterial population that's involved in the microbiome, but the genes that they're carrying and sharing, and what are the selective pressures for maintaining those, because they won't necessarily retain genes that they give them no selective advantage. So I think it's exciting times. Yes, the problem is getting good enough DNA to get good enough sequence to make the comparisons over time.
It's a simple question, but the dog had the most variety of different clones that I could see. And the mutation rate you talked about was 6 times higher than what they expected. Did they say that they thought that was because the dog got antibiotics? Like, I'm wondering, is that the reason why the bacterial populations in the dog were changing?
I think not, because the dog's infection resolved by the use of the antibiotics. So I've got the paper there. 6 times higher is just simply because there are some selective pressures, and the only thing they could speculate on was host adaptation. That the changes between the humans, things that are going through the dog and then reappearing in the human, says it's now not selecting specifically for advantage in the dog. But there's not enough mutations and not enough samples to make any sort of conclusion on what was driving the change.
Be interested to compare with dog that didn't get antibiotics and see, how the populations change over time.
Yes. So if they didn't give the antibiotics and the infection resolved itself, I'm not sure that it would be all that different. Clearly, there's just a lot of circulating bacteria in the family, which would be normal.
It's a bit, I'm just thinking about travelling and going overseas and drinking water and eating food and acquiring all this E. coli and coming back. And surely, even for that family, they've been mobile, they've gone places.
Gone to school.
How much, there's quite a lot of assumptions in there because it was over quite a long period of time, really. But I suppose they've just honed in on particular. That's what they've done, haven't they? There could have been a whole lot of other noise, but they've just...
Yeah, what they've looked at is that let's look at E. coli and let's see what clones persist. And the noise is what doesn't persist, right? And
maybe the dog, went to the dog park and ate a lot of other dog poo and, did other things to acquire E. coli rather than, just pick it up from family members.
yes, it could be the visitor that comes and kisses the dog. That's a problem. There's lots of inflows which you can't control for in that sort of an experiment.
Wow. That's an amazing study. Helen, say goodbye.
Oh.
Say thanks.
Beg your pardon? Goodbye.
Just apologise.
Okay, thanks everyone for putting up with the technical difficulties in today's presentation. It is because our two technical experts were away today, unfortunately. We'll have it back up and running next session. Thanks for attending, and we'll see you again soon.